So it's my pleasure to introduce now and return the favor. And the next speaker is Jeff Clogston, who is a principal senior scientist at the National Characterization Institute in the US, and also partner of the UNCL. And he's going to follow on my presentation on a more complex aspect. Jeff?
Thank you very much. So first, I want to thank Adriel for the introduction and thank everyone here for showing up. I get the honor of presenting and being moderator, so I guess I can take as long as I want, but we'll see. So like Adriel said, my name is Jeff Clogston. I'm a principal scientist and also the Physicochemical Characterization head at the Nanotechnology Characterization Lab back in the States. So what I want to do first is give you some background as to what we do at NCL. So NCL was established back in 2004 as a collaboration among NCI, NIST, and FDA with 4 primary objectives. The first is to characterize nanoparticles using standardized methods, to conduct structure-activity relationships, to facilitate regulatory review of nanotech constructs, and finally to engage in educational and knowledge-sharing efforts like I'm doing here today. Now, we have what we call an assay cascade, whereby when particles are submitted to us by government, pharma, academia, or biotech companies, these go through an assay cascade where we can characterize some things for it. For example, sterility, immunology, pharmacology, PCC, toxicity, and efficacy. We then use this characterization basically to support their developmental, clinical, regulatory, and commercialization needs. And what I should mention here from all this thing, it's actually a free service. One just has to apply through us through a white paper process. Now, I mentioned this assay cascade. We have 3 tiers to this. There's PCC, of which I'm in charge of. And what I've listed here, and I won't go through this, basically is the parameters that we can look at, along with some of the techniques associated with each of these parameters. We also have an in vivo characterization. So we can look at sterility, hematology, toxicology, to name a few. And then in vivo, so this is your immunotox, your pharmacology experiments. And the idea behind this really is to link the PCC properties to some biological outcomes. Now, I mentioned I'm part of this group, so the rest of my talk now, I want to kind of focus on the PCC side of things, because really that's what we want to talk about today. So within PCC, if you think about this, really, PCC comes down to your analytical instrumentation and development of new methods. So I have listed here what we have in-house at NCL. Now, based on all these instrumentation that we have, we're fortunate to have a lot of these different tools in our lab. What we've done is come up with all these instrumentation, and over 12 years experience, we've been able to identify the critical quality attributes and methodology needed to characterize, and I listed here just the 3 main platforms of liposomal products, polymeric nanoparticles, and colloidal metal nanoparticles. Now, I'm not going to go into detail with all these, obviously, but if you're interested, I actually have come up with the cheat sheet, so to speak, that basically goes through all the CQAs for each of these 3 platforms. It gives examples on what to look for, what to ask, what you should ask in characterization, and how to do it. So if you're interested, come see me after. But what I want to do now is spend the rest of my talk is to look at one critical quality attribute, namely sizing, and that's what we're here for today. So I will go through some examples in our lab where we've characterized our client particles and just show you examples on how we characterize and what techniques. So the first one I have here are for lipid protein nanoparticles. So we measure this by dynamic light scattering. You can see some particles are out here. We all know that DLS is not suitable for above micron particles. We did laser diffraction. We can see the smaller peak here, but now with laser diffraction, we can see this presence of this larger size population. Using AF4, asymmetric flow field flow fractionation, with MOLs, UV, and DLS in line, we can further separate the particles out from any free protein as well as lipid aggregates. So the take-home message here really is that orthogonal methods are highly recommended, and Adriel actually pointed to this early on in his presentation. And I think most of us here know this already, to use many different ways to look at one parameter, sizing in this case. My next example is Cryo-TEM. So this is PEGylated liposomal Doxorubicin. Most of us are familiar with Cryo-TEM. We can see morphology and shape analysis here. We can see the crystal inside that crystallizes. Based off of this micrograph, we can actually measure diameter. From that, you can calculate the particle volume. You can also measure the bilayer thickness if you need to do this, calculate inner diameter, inner volume, things like this. This is pretty straightforward for liposomal work. We can also look at particle size and concentration. So this is a technique using resistive pulse sensing, and this is Spectrodyne's instrument, where we've looked at particle size and concentration for 3 liposomal formulations. 2 are here. The first 2 are actually just liposomal doxorubicin from 2 different manufacturers, Doxo and Sun Pharma. From this, you can see basically the average size range as well as the concentration for these 2 samples. Included, we looked at irinotecan liposomes, AuNPS in this case, and you can get similar analysis for this. So this is tunable, well, this is resistive pulse sensing. We also have tunable resistive pulse sensing, which is our Q nano, our eyes on Q nano. And in this case, we actually used this to look at batch-to-batch consistency of 3 different lots. And we were submitted 3 lots of silica core gold shell PEG-coated nanoparticles. We ran this through the Q nano. So in addition to getting size as well as particle number, you can actually look at the charge distribution using this instrument. And that's what's shown here, just a scatter plot of zeta potential as a function of size for all 3 lots. So using this technique, we can actually look at the differences between these 3 batches more so than, let's say, a DLS measurement can. The next one is using single particle counting ICP-MS. This is relatively new. And what we did here, proof of principle, is we took a mixture of 40, 60, 100, and 150 nanometer gold colloidal gold nanoparticles. We mixed this, and then run this through the ICP-MS using single particle mode. Using a calibration curve based off of NIST SRMs, and using we assume it's a sphere, of course. Most of our nanoparticles are sphere. We make that assumption. Using this, we can actually back calculate the different size populations in this sample in a single run. And if we compare this against the measured TM sizes, you see there's actually very good agreement between the 2 methods. There we go, okay. My next couple of slides actually deal with AF4, so I find this to be a very strong separation technique. And again, proof of principle, so we took a mixture of 30 nanometer colloidal gold nanoparticles, and what we did, we grafted different peg lengths. So in this case, 2K, 5K, 10, and 20K. If you put this together and do a batch mode DLS measurement, you see 1 peak. Obviously, DLS in this case is limited in the resolving power. However, if you hook this up to an AF4, which can separate the nanoparticles, and have inline mols and DLS, what you get now is that you can actually baseline, well, not baseline resolve, but you can resolve the 4 different species in the samples using mols detection as well as DLS. Now, this is a proof of principle example. Let's look at a real-world example. So here, again, using AF4 with DLS, we had 1 client submit to us dual drug-loaded liposome. So if you did batch mode DLS, you see the sample is polydispersed. If we run this through AF4 with DLS, we can actually tease out the 2 different size populations. And here, you're getting information now on constant size regions as well as increasing size regions in the sample. A better example is a micellar SN38 formulation. So if you look at batch mode DLS, you can see here, obviously, it's polydispersed. You can pick any number in here, and you'd be right with the size. If we run this through AF4 DLS, we can actually resolve the 2 populations shown here. So this more represents what we do in-house pretty much all the time now. I like DLS, but for me, batch mode DLS is very limited in that case. So having some sort of separation upstream and then having DLS as one of your detection methods is very useful, as you can see here. Another example with AF4, again, going back to this back-to-batch consistency, this is very important that you should look at this. So our client supplied to us 3 lots of PEGylated metal oxide nanoparticles. We initially did TEM to look at this. Now, if you look at these 3 micrographs, can you tell me if there's a difference here? Probably not. And if you look at the numbers here, there is no difference. So what we did was basically take this and run this through AF4 mols, and that's what's shown here from the chromatogram. You can see 2 lots are very similar. 1 lot, lot 1, is very different in terms of size distribution. So again, using this technique as a way to assess batch-to-batch consistency. My last example, before I move on to my last topic, is to assess protein binding. So this kind of hot topic, so to speak. And what we're doing here is we're using AF4 in conjunction with mols and DLS detection methods. And what we want to use here is basically to see if there's any protein binding to a nanoparticle. So our proof of principle experiment here was to look at 3 commercially available formulations. The first, Doxo. And I want to make note here, this has 5% PEG, lipid PEG on there. If we look at AuNPS, there's a little less, 0.3. And then finally, Ambisome, which has no PEGylation. If we now take each of these and run this through a system with and without incubation with human plasma, what we see is the following. So for Doxo, I'll show you here in in blue is without plasma, in red is with human plasma. You can see AF4 can actually separate the free unbound plasma proteins from the nanoparticle. And what we're looking for here is a shape factor, or this ratio of Rg over Rh. This basically gives you indication of mass distribution on the sample. So in the case of before, this ratio is roughly 0.77. After incubation with plasma, 0.8. So not a big difference. So this implies qualitatively there's no protein binding to the surface. If we now look at Onivide with little less PEG, and we run the same type of analysis with and without plasma through this system, you can see the shape factor actually increases slightly, right? So there's basically some protein on the surface of the liposome. If we look at ambisome now, this number goes almost 50% increase, suggesting that there's protein binding to the surface. You actually don't even have to calculate this. If you look at the fractogram, you can tell there's definitely something going on here. So what we're doing is basically it's a way to look at size distribution, as well as protein binding using AF4 mols DLS. We're currently working with QCMD, quartz crystal microbalance, with dissipation to actually quantitatively assess how much protein is bound on the surfaces. So what I want to do now is spend the last couple of my minutes that I have left is just kind of update you on a meeting we had 2 weeks ago at the ACS meeting in Boston, USA. And same title, actually. We kind of modeled this workshop after this one. And on our particle sizing team was me, as well as Patrick Limso from Pfizer, and yours truly is also here as part of our committee. And what we did was we put together a nice workshop. It was a 4-hour workshop where we invited people from first regulatory, from FDA, from Metrology, NIST, and then we had several pharmaceutical companies, Merrimack, AstraZeneca, and Pfizer. We then had invited some instrument vendors as well, Malvern, Spectrodyne, and Manta. And really the idea was to give us the metrology and regulatory aspect of sizing nanoparticles, to give us how it's used in industry, and finally we gave the vendors a chance to introduce some of these newer technologies that's coming out there, beyond DLS, for example. Finally, we had a panel discussion led by the 3 keynote speakers where we asked questions. And my last slide really is what came out of this meeting. So from that roundtable discussion, 3 topics came up, and I'm hoping that maybe we can use these topics as a stepping stone for our discussion later on during the Q&A part or the debate part of this session. So the first one had really to do with when you measure samples, for example, in DLS, with DLS, do you just measure it in buffer, or do you actually measure this in human plasma? So we posed these questions to the audience as well as the keynote speakers, and they had very interesting results. The question I always have is, do people do this because they're not cognizant of it, or second, because it's a hard issue to do? It's an instrument limitation, for example, or they just don't want to do it because they're scared of the results. So that could be a very contentious topic there. The second one, and this was obviously directed to Debbie Kaiser from NIST, is what are NIST priorities with respect to development of RM? So we all know there's colloidal nanoparticle RMs out there. So the question is, do people actually use them in their methodology, for example, to test their sizing protocol? Second, are there any plans for particle count reference materials? I know Adriele brought this up, and I know a lot of people in the audience might bring this up too, is if you have techniques that do both size and concentration, what are the RMs available for particle count? I know they're not working on this currently, but that's something we can discuss later on as well. The last one, and I actually asked this question, so Celia Cruz actually presented a paper that was recently published by Cedar FDA where they did all these statistics and what have you. And most of the sizing techniques out there are based on dynamic light scattering, laser diffraction, and microscopy. Now, with the advent of all these new emerging sizing techniques, how is FDA going to handle this? Are they starting to see a shift where people are using, besides DLS, other methods, or are they just kind of basically dropping off DLS and laser diffraction and only using the newer techniques to look at their particle? So that's a question I had to them, and that's something they're still working on as well. So I'm hoping that with these questions, along with any questions you guys have, we'll have a good, lively debate later on. With that, I want to thank the NCL team and thank all you guys for your attention.
Thank you, Jeff. So as you see, we try pretty much to actually mirror what happened in Boston, so we can actually draw up a US-EU parallel discussion. And we're taking advantage of this morning to actually seed the pitch and set the ground And then in the afternoon, in the second part, to actually have an open discussion where we have a back and forth, and we can collect all this. And the idea that we have behind the team is to actually develop into a white paper in the foreseeable. So any question for Jeff?
I would say it's probably the most popular because it's been around the longest, and it's probably the easiest for someone with an instrument. Just, well, you're familiar, and most of us are familiar with DLS instrument measurements. You take a cuvette, stick it in, grab coffee, come back, and you get a number, right? So I think it's been around for a long time because of that reason. People still use it. I know there's other techniques coming out there, hence my question to FDA at the roundtable discussion at the ACS meeting, because there are a lot more, as you know, as well as most of us know, there's a lot of new techniques coming out there that can do a little more than sizing for us, and probably better. Any other questions? Okay.
If not, Jeff, thank you very much. I'll give you back your job.
Okay, thank you. So I'd like to introduce